he also worked on this area, but he has found that, between these repeats, they are called spacer, which come from the virus, and that's this virus, was virus fragments coming from the virus, that's he just discovered. And then the other one is Radolf Barango, is a food scientist, when he was working in the yogurt, he just found it, and then he has published one paper, in this area, okay? And then this Jennifer Doudna and then Emmanuel Carpentier, that is two lady scientists, and then by the paper that they got it 2012, and they got the Nobel Prize. But there are two groups at the moment, at that time, working on this area. One of them is the Jennifer Doudna groups in Berkeley, and the other one is the Feng Zheng group in the MIT group, But actually, the Nobel Prize has gone to Jennifer Doudna and the friend Emmanuel Carpentier, but the patent has gone to Feng Zheng, The problem is that, these two scientists, two groups, working on this area, but the beginning of this one was due to the Jennifer Doudna, just found it firstly, and then after that, Feng Zheng, with the MIT group, is a published, application of this CRISPR in animal cells, eukaryotic cells, okay? And then what Jennifer Doudna found that, Jennifer Doudna just only found at the beginning, the defense mechanism of bacteria, and the MIT group, then founded and applied a patent, for the application of the CRISPR to the eukaryotic cell. It means that only to find the mechanism of the defense mechanism is not enough, because it has to be applied to the humans. That's why this is important. And then there was about the Jennifer Doudna has also found that the application of this CRISPR to the humans, but it was 2 months or 3 months later, okay. And then now there is a fight between these two groups, and then one is very important thing that I want to tell as a scientist, young scientist, you are, they put their claims on the notebooks, experimental notebooks. And what did the Jennifer group say that, they had done this and they had shown that this is on the notebooks. So it means that when you make a scientific experiment, please do always notebook, always notebook. This is a proof of something later on, Well, this is all about the story of CRISPR, but there are a lot of things to be said more. Okay. Now, as I said before, that the Ishino first scientist that, Japanese, and he has found only the repeats, palindromic repeats. And then we have to put in our mind that always whenever we talk, repeats are in loop shape, okay, loop shape. And this is important to remember it. So let's go to the other one. Okay, now, how come this happens, Okay. This is let me call this is a red virus, or red virus. And then when injects to DNA to bacteria, and then what happens, bacteria has a defense mechanism, and what is, as you see that this is double-strand DNA and this is viral DNA, okay? And there is a complex system in the bacteria, what is called case 1 and case 2 system, they cut it, as you could see here from here, and put the CRISPR array, What does the CRISPR array, it means in bacteria. In bacteria, as you could see, there was repeats, RRR repeats, palindromic repeats. And as you could see that this was blue, and this is also purple, This is a spacer, This has been taken by about maybe last week or 2 years ago. Okay, there are about fragments of DNA in the bacteria, okay, like what we call this spacer, And then when this double-strand DNA has been taken and put it here, and this step is called spacer acquisition, spacer acquisition. Okay, now what happens next time, Then, as you could say that this is the CRISPR array, this is spacer, which these are different color is due to different virus fragments, and the repeats is okay the same one, and repeats are the same in palindromic forms. Okay, now what happens, when the first time comes to the red one to the system, then the bacteria tries to make, process this DNA as an RNA, Okay, so it means that this whole system, the DNA converted to a single RNA system, Okay, then as you could see, that repeats is palindromic, and then these are the fragments of a virus. It was first DNA, and then it's converted to RNA, And again, the repeats are the same, and then this was the DNA first, and then it's converted to RNA. And this is repeats and spacer, repeats and this spacer. And then, what we call this one, this is biogenesis and processing step, okay? So it means that the CRISPR transcribe into one long RNA molecule. This is, okay, RNA molecule, and then into shorter pieces that is contain single. Okay, then in that case, after this RNA, then it is cut it down, and as you could see from the here, then what you get, for instance, this is the protein part, and this is the virus RNA, and this is CRISPR RNA. And then this is, okay, this is the blue one, and this is, okay, dark blue, So it means that when you get this one, this is what we call CRISPR RNA, and on the surface of the protein, Okay. So since these are the Cas9, so we can call that Cas9. So this was the memory, and then what happens? Let's come to the interference system. Okay, so what we get finally, this is the CRISPR RNA, which is made of from the fragments of virus DNA, but it's now is single-strand RNA now. Okay, so this will be element that recognize the target DNA. Okay, so when the infection, when the second time the virus infect the bacteria, and then the bacteria, what we have it, this CRISPR RNA, that is red part, and this is repeat part. Then since this was originated from this red DNA, then this recognizes this one. When this recognizing this one, and the Cas9 enzymes cuts down the double-strand DNA. So this is what we call, as you could say now, the DNA of the virus has been killed out. Okay, it means that this is the virus has been killed out. So this is the way the defense mechanism of the system. Okay, now the problem is now this. Okay, what does it bring? This doesn't bring anything else. That it has to be applied to for instance, the clinically, or applied to animal cells, to human cells. Okay, so what we have learned now that the in this case the CRISPR defense mechanism, as you could say, that
the foreign DNA has been taken by the bacteria and then put it in the as a spacer, as a DNA, and then it's converted to RNA, and then finally they have got CRISPR RNA. Okay, so this is the first thing that has happened. Okay, now how come this name of the CRISPR has been given? The two scientists, the Janssen and the Moika, these two scientists, one of them is Janssen from Holland, and the other one is the Francisco Spanish, and this is the name has been given by these two scientists because this is important, and then it is given. And one of the story about this invention that Moika, when the first time after he has got the PhD, he returns back to university, but he had no funding, so he had no funding. So what he did, but he has only one computer. So what he tried to find it, he find a computer that this between these repeats, he searched these repeats. Between these repeats, the DNA fragments coming from the virus. So he just found this one as a recognizing element. So next one, I will be giving this one. Well, just as you know that you already know, I'm just passing very quickly that the nucleotides of DNA, is made of phosphate, sugar, and nucleic bases. This is because someone who wants to work with the CRISPR has to know exactly what is the DNA, okay, and that's very important. And the structure of DNA, as you can see, phosphate and the sugar and the primitive bases. Well, why do we call these bases? Do you have any idea about this? The bases, adenine, thymine, cytosine, guanine, okay. So why do we call this one the bases? Well, these bases, all of them contains nitrogen, and nitrogen has, unshared pair electron. And this means that it can join with the proton. So it means that it's also called as a base, That's why, when you talk about the base, it means nitrogen is base. And this is why it's called the base, because it has the nitrogens, that is, nitrogen has two unshared electrons. And due to this unshared electron, it's called the bases, okay. And this is the phosphate. And the important thing that you have to work in the future, that if you want to work the electrochemistry of the DNA, that is the negative sign. So it means that the DNA is negative signs. If you buy some DNA from a market or any case in the companies, that is negative due to the phosphate group. So. But that how come this compensated, okay, that all we have is natural, natural. There is no zero sign, It means that the proteins which has the positive part and this is the negative part is compensated. Okay, this is the important part. And then this is the bond between the phosphodiester bond. And as you know, the nuclease, this Cas9 or Cas12, is the enzyme which cuts down these bonds. It means that between two nucleotides, that's cut it down. This phosphodiester bond is been broken. Okay, please remember that. Whenever you talk about the Cas9, Cas12, and Cas13, it is these enzymes can cut down the phosphodiester bond, okay, between two nucleotides. Okay, this is very, very important, basic things, okay? And what is the difference between DNA and RNA? In sugar, if there is a hydroxyl groups in both of them, two prime and 3 prime, then it is called ribose. And that's why we call it ribonucleic acid. That is RNA, you know. And if there is no oxygen at 2 position, you know, that's call it deoxyribose. And please remember that, you know, this is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 4. But it's called 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. This is very, very important to know it, okay? The basic things. And the sugar was the pentose, okay, the sugar ribose. Well, the central dogma is this one, and that's okay. There are hydrogen bonds between these bases, you know, adenine, thymine, cytosine, guanine, this basic principle. And then the central dogma is this one, DNA, RNA, and the protein, you know. As you could see that the DNA converts to RNA, this RNA, and then goes to protein. Okay. Now, this is, in our case, in animal cell, in human cells, only 2% of our DNA goes to protein. What about the 98% of it, you know? Instead, only 2% of our DNA goes to protein, and the 98% of our DNA stays as RNA, you know, okay. Okay, this is the one which goes to protein, and it goes to coding DNA, and which the other one is non-coding DNA, RNA, sorry, okay. So this is the some basic principles. And again, you know, the timeline is the first one was the Ishinawa, okay, and then Moika at all 2000, and then the 2007, you know, this was the Barango. This Barango has a chief editor of journal, CRISPR journal, and it's very good journal that you can follow it. And then this is the group of the Daltna, the first author, Genek, and then the other one is the group of MIT group. These are the main milestones of the CRISPR technology. Again, you know, main scientists, the Feng Seng group, MIT group, and Jennifer Doudna, and then the Emanuele Pinter. And I would like to give you one example for the story of this one. Jennifer and the Carpenter, one of them is United States, the other one in France or in Germany, you know. When they joined together, you know, one meeting, you know, they began to cooperation, you know, okay. And this is very, very important for also for a scientist to communicate with different scientists. This is must, you know, you cannot do everything by alone, you know. You should contact with your colleagues at the same topics, you know, that in that case you can have different outcomes. So 1 + 1 is not equal to 2 when you make a cooperation, okay? Then please remember that always try to cooperate with other scientists, okay? 1 + 1 is not equal to 2, it may equal to 3 or 4, you know. Okay, this is very important. Okay, now this was the main scientist. Okay, this is, as you know, that. I had the chance to listen to Emanuele Carpentier before the Nobel Prize, you know, when I was in USA. She gave a conference in Columbia University in New York City. I was one of the listener. Okay. So now, okay, now let's go different explanation of how the defense mechanism of the bacteria. As you could see that, you know, when this time, you know, the virus is coming, the blue virus, you know, okay.
when it comes down to, that double-strand DNA is put it as spacer, okay? And it is converted to RNA, and then this RNA has been cut it down, and together with the Cas9 enzyme, and then what we call it finally that, the enzyme plus guide RNA, you see, this is the most important part. Okay, when the same virus comes next time, as you could see that the one that is the blue one was originated from the blue virus, when this recognizes, matching, what do you mean by matching, the adenine thymine cytosine guanine matching, so it means that this is this triggers the enzyme, and it makes double strand breaks. Okay, so this was all about the defense mechanism. Now let's go to the other one. Okay, so there was Cas9 enzyme. Cas9 enzyme has two domains. One of them are UVC, and the other one HNH. Okay, and then as you could see that there are two RNAs in defense mechanism of the Cas9. This is one of them is the CRISPR RNA, and this is tracer RNA. Okay, now when as you could see, these are separately, in the defense mechanism of the bacteria. But also, what happens that when they join together, they synthesize this CRISPR RNA and tracer RNA in the laboratory. In this case, it's called single guide RNA. Okay, what is the difference between single guide RNA and guide RNA? The single guide RNA means that these are the synthetic guide RNA, which is made of tracer RNA and the CRISPR RNA. But I will be talking about the Cas12 and Cas13. In these enzymes, there is only one RNA, but in the Cas9, there is two RNA, tracer RNA and CRISPR RNA. Well, this was the paper that Jennifer Doudna. Okay. So there is a classification, CRISPR-Cas enzymes, class 1 and class 2, and most of them, this class 2 is this Cas9, Cas12, and Cas13, These three of them is very, very important, okay? This is three of them is important. As you could see, the target will be the Cas9 DNA, Cas12 DNA RNA, and Cas13 is the only RNA. Okay. Now, well, this is the application to humans. This is the gene editing. Okay. So what we have at the moment, we have the Cas9 enzymes. Okay, we have Cas9 enzymes, and we have a target gene. So what you want to do, that I would like to talk about what is mutated gene. What is mutation? Well, if someone stays over many times, on the sun, the sun, when it touches to the skin of the people, then what happens that the cancer happens, Actually, the gene of this skin, changes from, for instance, you have a adenine thymine cytosine. Okay, then adenine changes to guanine, for instance. This is when this A changes to guanine, the different bases, then this is a mutation. And these mutations cause the cancer, The reason of the cancer is due to change in the DNA sequence of the gene, Okay, that's very important. Now, So what we can do with the application of the CRISPR in gene editing, So this is the target sequence. So you want to have a gene, you want to have a gene and you have you want to knock out, okay? So it's something like, you want to kill down the virus, So take it analogy, virus and the virus DNA and the mutated DNA, okay? You want to eliminate this DNA because this DNA is mutated. It may cause the cancer. So how can you do this one, Okay, this is the problem. So you must have the sequence of DNA. Since all human genes are known, that maybe this is the X gene, X DNA, and then you want to knock out this gene, So what you have to do, first of all, you have to have the guide RNA. As you can see, the guide RNA is at the beginning, it repeats and then spacer, okay, together. But now, instead of the spacer, this is the part of the guide RNA which will touch, which will bind to the target sequence. When this binding this one happens, then this Cas9 will be, activated. It means that these scissors, you will see that will cut down. Okay, so what happens? This is the double-stranded DNA is broken down. So what happens in this case? Well, one question to you, Now I just explain you. Defense mechanism bacteria. What happens, when the second time the virus came, and then what happens in this case? The virus has been killed down. Okay, but when we talk about the human system, that systems, there is no way that you can kill down the double-strand DNA. So you break down this double-strand DNA. It was something like that, and you cut it down, But as you know that in humans, that when you cut down something, they want to join together. Repair mechanism is involving, repairing the DNA. It was already cut it down, and they want to join together. It's a very important question. When they joined together, does rejoin from the same place or not? They are not joining from the same place. Why? Due to reasons that, some of the DNA, for instance, this is the original DNA, as you can see here, okay, that's the double-strand DNA, has been, deletion. Some of the DNA or bases are deleted, and some of them, as you can see, green one inserted, This is what we call indels. So it means that due to deletions and insertions, what we call indels, then when they join together, they're not the same DNA as before. So it means that what happened that, you change the DNA of the system, and then since this is not the same DNA, it's not the same gene as before, it means that it will not form any protein. So it means that this will be eliminated. So what we call this process, knockout, KO, KO. Okay, so this is when they join it this way. What we call this one, no donor template, non-homology and joining. So it means that by itself, if you let it join together, and this joining, will not be the same as the original one. Okay, so what the second procedure that you can apply, is a gene editing, since this was cut it down, you can add a donor template. Okay, this is homology directed repair. This is It means that there are two repair mechanisms. One of them, non-homology chain joining. It means let it join by itself, okay? By joining them together, they are not joining from the same places. So it forms a different DNA, and different DNA means the different DNA, different gene, and this doesn't happen any protein. So this was knockout. This is the one way, and then this technology will be used for other purposes, okay, for different purposes. Please put in your mind. Okay, now let's go to other ones. Okay, so I will be talking about this one. Okay. Now, if you have any question, this is the first part of this one. After 5 minutes break, I will be, explaining other parts to you. Okay. Let me have it. Okay. Where's my? Okay. Okay. Yeah. Hello, everybody. You hear me?
Yes,
Dr.
yes,
Muhtar? Yes. Okay. Do you have any question? Let me. Okay.
Students, do you have any question to Prof? Okay, Ian put there have no question at the moment.
I will have 5 minutes break, okay, or about 5 or 6 minutes. Then I will join it, and then I will be writing down. Okay.
Okay. So break now, huh?
Okay.
Is it break
Yeah,
now?
break now. It's
okay.
okay. Okay, it's about okay. Okay, we'll meet you in 5 minutes. Okay, see you.
are you able to see my web table? Yes.
Actually, there is no words.
Okay, I can begin now. Okay.
Okay.
Okay. Welcome everybody. Again, I just explained the things that I've done. Let me know it. Okay. Now I will be doing the figures for your case. Okay. Now I'll explain again what is the defense mechanism. Defense mechanism is of CRISPR. Okay, CRISPR. So okay, black one. Okay, so this is the bacteria. In bacteria, what we have, repeats, pollen repeats. Okay, and then what happens? Proto spacer. Okay. When this comes to here, then it becomes spacer. Okay, spacer. This is very important. Okay, now the one thing that is very important, how can you cut down? Case 1 and case 2 is cuts down from here and put it here. So what happens in this case? This is the repeats, and then this DNA converted to RNA, RNA, and then this is called what we call it. Then this is RNA, and the black one. Let me show you. This is the black. Is this the okay? And then this is again the black one, and maybe the other color. This is the green one. Is the another RNA, and this the black one still. This one. Okay, this is the RNA. So it means that this is the red virus came it and then as a DNA and it's converted to RNA. And then what happens in this case? This is cut it down from here, and then this is cut it down from here. In that case, what we have with this one. As you could see that. Let me show you this one. This is the red one. This is coming from the virus, and then black one is coming from the from bacteria. And this is the repeat. So this case for the CRISPR RNA. Okay. Well, if this is when the virus came to the second time, let me go this one. Okay. Let's see. How can I change this one? Okay. I will be okay. Another one. Okay. So let me clean this one. Okay. So. Okay. Now this is the CRISPR RNA that we get it. When the okay now. Come back. Okay. Okay. This is the when the this is the bacteria, and again the bacteria comes to a second time. The virus comes to second time. Okay. So when this virus comes the second time. Okay. And this is the CRISPR RNA. This has already been obtained, and then this is the black one. Is how can we show this one? Okay. 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 So when this match this one, what happens in this case? The enzyme, what we call it Cas9 enzyme, Cas9 enzymes activated and then makes double strand breaks. Okay, double strand breaks. So then when this the second time virus came to the bacteria, and then the virus has been killed down. Okay, this was the first thing that has been done it. Okay, now let's go to not possible. Okay, okay, okay. In this case, let me show this second time. Okay. 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 Sorry for that. Share again. Sorry. Okay. Whiteboard. Okay. Okay. Now I come back to again. That we have discussed the Cas9 enzyme. Cas9 enzyme. Okay. What does he do? There is two RNA. Two RNA. One of them CRISPR RNA, and then tracer RNA. Okay. Okay. When they join together, they call it single guide RNA. Okay, single guide RNA, and this is for Cas9 enzyme. But when we have it Cas12 enzyme, okay, then we have only one CRISPR RNA. Then Cas9, Cas12 can cut it can cut the double strand DNA. Okay, but what we have with Cas13, it can cut down the RNA. Okay, now so this is the basic thing for Cas9 enzyme, Cas12 enzyme, and Cas13 enzyme. Okay. So now I will be passing to, okay, 1 second. I will come back to the original one. I will be shut down and then I will come back again, Okay. 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 So, I'm back here, sorry for that, miss problems due to miss problems. Now I'm sharing, okay, again. Screen, yes. Okay. Apple Store screen, okay. Yeah. Yes. Now I'm back here, yes. Now I will be showing this one. Okay. Now everybody can see my screen now, and then I will be okay. Now, if you have any problem, let me know it. Okay. Now I will be talking about the Cas9 enzyme that I already discussed it. This one, and Cas12 enzyme and Cas13 enzyme. Okay. And this is the single guide RNA, as you can see, that's the blue one. And this is the yellow one, is complementary to this target DNA, okay? And there is another important thing that is very important for electrochemically, nickase, nickase Cas9. there is only one Caesar in Nikkei's case 9. Okay, this is clear now. And then when we come back to case 12, this is very, very important. Please remember this for analytical purpose. If you want to design a biosensor electrochemical, that you have to use this technology. Okay, now this is the double-strand DNA, a blue one, and then the yellow one is the spacer, part of the CRISPR RNA, and then when the target comes hit, it can cut the double-strand DNA, as you could see here, but at the same time, anything around next to it, okay. Well, it means that Cas12 cuts double-strand DNA, but at the same time, it can cut any single-strand DNA around it here, Okay, so it means that Cas12 can cut any single-strand DNA around it, But if this is a reporter, then if a reporter, then you will see the light of it, And Cas13 is the same thing, okay? Same thing. Now, let me go to other ones. Yes. Well, electrochemistry of Cas12, Okay, this is a Cas12 enzyme, Cas12 enzyme. And then this is the target, as you could see, that blue one and the red one, see. When the CRISPR RNA matches this red one, then it can cut the Cas12 target DNA, but at the same time, it can cut any single-strand DNA around it. It can cut it. Okay, now how can we apply this electrochemistry? Okay, if there is no target, it means that there will be no cutting. Okay, the non-target. So this is our electrode system, the gold electrode, and this is the single-strand DNA, and this is metal blue. Metal blue is a electrochemically active element, So what happens in this case, if there is a target and if there is no target, okay? As you can see that there is no, this is the CRISPR RNA, see, and the CRISPR RNA, and as you can see, this is the target. When there is a target, it can cut any single-strand DNA and it cut it down. But in this case, there is no target, and this is still here, It means that without target, in this case, without target, you get a high response, but with target, you get a low target. So this is very, very important. In future, you want to work with electrochemistry, and you can go to my papers also. You can have it very clearly. Now, the same thing applies. This is Cas12, This is CRISPR RNA. When there is a target, target DNA, then it can cut it. Not only the DNA, but it also cut any single strand around it. So it means that it can cut the single strand DNA, and then this is with target and without target. Okay. Okay, now very important. So this is Cas12. Okay, this is a new paper, has been published, This is Cas12 enzyme only, and this is guide RNA. When they join together, Cas12 guide RNA, so it means that it can cut any target that is already there, So when this target DNA matches the guide RNA, see, this is two scissors. So it means that it can cut the double-strand DNA, but at the same time, any single strand around it, okay, single strand. So we can use this technology for application, For instance, CRISPR-Cas12a, these are the loops, loops. And when as you could see that when there is a target, then it can cut this one, and the response when there is target low, and when there is no target, this response is higher. And then, and this is very important thing, and then this paper has been published already, how can you detect the coronavirus, okay, COVID-19 nucleocapsid protein. This is the COVID-19 virus. It may have it, there are spike protein, as you could see, and the membrane protein and amyloid protein. And this is another one, nucleocapsid protein. And I will talk about and what is the aptamer. Do you have any knowledge about the aptamers? Aptamers are nucleic acid structure, which with structure which can bind to protein. Okay, it means that aptamers can bind to protein. Okay, then this is the nucleocapsid protein. And this red one is what we call it, the aptamer. But at the beginning, what we are giving to the system, double-strand DNA. One of them is the aptamer, the red one. The other one, the activator strand. What is the activator strand? I will be showing you up there. So when the aptamer, which is protein, when they act to this molecule, they can attract this red one, okay, and separated one, and then the green one is free, and this free activated strand joined with the Cas12 and then can cut the methylene blue. But in this case, if there is no protein, it means that if there is no COVID-19, it means that there will be no cutting business, and that means that the control will be higher, So it means that this way it can be detected the coronavirus. This is really, really interesting. So what is given here, aptamers is a DNA structure which can bind to the proteins, It means nucleocapsid protein can bind to red aptamers. So this is another topic that is very, very important for your case. Maybe you have seen this aptamer in your study. So this is how we can do it with electrochemistry behind it. So, okay, this one. Okay. Okay, now I will be giving, this is one thing. Yeah. And let me see what time you said we get 11. Okay. So this is a paper that we have done it, okay. Electrochemical sensors. Well, this is a gene. Okay, what is a gene? Is a cyclin gene, This is used for detection of the cancer, So, this is the DNA of the gene, okay, gene. And then you want to make double strand breaks. So what you do, you add this CRISPR RNA to the system, and then when you add CRISPR RNA, as you could see that it is double strand breaks happened, okay. So it means that we have a double strand DNA. So how can we do this one? There are two ways that I meant it, non-homology and joining. That means that it can join it, but then at the end, what you get it, you knock out. But in our cases, what we have done it, we add, point mutation to the system. So it means that we created, mutation to the system, and then we are trying to find that how come this happens, So this is another technology we have done it. Okay. And then what else I want to show you more? Okay, Pango. Okay. Okay, now everything is okay for the CRISPR, but now the problem is this off target, See, what is off target, It's very important in this case in CRISPR technology, for instance, you have a book, in this book, you want to look a word, for instance, nice, nice, but when you make search, you can also search the word nicer words. So it means there's a mistake, Okay, this is due to this mistake, joining of this CRISPR, is maybe difficult, So it means that the CRISPR technology cannot be applied to embryo state, embryo state. This Chinese scientists, make experiment on the CRISPR system on embryo state, In embryo state, this is not possible to make it, So, and due to this experiment, the Chinese government put him in the jail, It means that you cannot do experiment on the embryo level, but you can do the experiment on the maybe lung cancer treatment, hepatitis cancer treatment, but not the embryo state. What is embryo means that the baby about 1 week, 2 weeks, this is what we call embryo. So you cannot apply the CRISPR technology embryo level, Okay, so this is important to say it. Okay. Okay. Now, how can we apply this is application of the CRISPR in sight loss, Okay, the progression of Stargardt disease. What is Stargardt disease? There is a gene, what we call this is ABC gene. This is fault, As you remember that DNA goes to RNA and then goes to protein. This is what we call central dogma. If the DNA is right DNA, no problem that you can get the protein. But if the protein is faulty, then DNA is faulty, then you get the faulty protein, So it means that the gene, ABCA4 gene, was a problem, Okay, this is a fault, It misgets. How can you eliminate this one, So by CRISPR technology, you can apply this one, and then you can eliminate this gene, and then the disease has been solved this way. Okay, it means that the patients with has sight loss by application of CRISPR, it can be impaired, Okay, this is very, very important. And then if you want to go to this topic, you can go on this paper. Okay. So this is really one of the application of paper. It's about 4 months only, and then you can correct the sight loss of anybody. Okay. Okay. And this was, I talk about it. Okay, now what else I will be explaining to you. Okay. Okay. Now, one of the important thing that I want to tell you is the off target, So, sorry. If you want to learn more about the CRISPR, one of the main important thing is the off target. Okay. So this is your DNA DNA. Okay, DNA. Okay. And this is your guide RNA. This is guide RNA. And as you can see, the guide RNA, this RNA is, this is A and T, G and C, and this is, okay, this is complementarity. Okay. Okay, and this is in target, you know, there is another cases, NGG, you know. NGG is protospacer adjacent motif, you know. Protospacer adjacent motif, you know. Before the spacer, you know, it's called it, you know. This is your target DNA. And as you could see that, you know, the matching, you know, here can match it here, but it can also match it here, you know. Why this happened, you know, by mistakes, you know, this can happen. So it means that, for instance, this is TTT, and instead of the, you know, the C, you know, it may happen that this could be T, you know. When this guide RNA matches to the target DNA by mistake, you know, you can have more than one places, you know. Okay, it's one more than one place. So it means the enzyme can cut not only here, you know, but it can cut here, you know, more than one places, you know. So it means that why this is not possible, you know, to use this for embryo level, you know. So no one knows what happens, you know. Well, it may be more than one places, it may cut it down the DNA, and this may be, you know, give us a problem, you know. So it means that if you want to continue on the CRISPR technology, should put you in your mind that off target is very important thing, you know. It's very, very important, you know. So what I said before that, you know, how come this technology precision, you know, is that I said to you that if there is a 1 million words in one book, you can search that word, you know, what you wanted, and you can change this way. But at the same time, you know, this is a big problem. Well, there is another technology that is developed later, you know, base editing and prime editing, you know. Okay, now what you have learned more in that just recently, you know, recently, this is base editing and prime editing. In this case, there will be no cutting situation. In this case, there will be a very good opportunity to use this technology. Okay, okay, base editing and prime editing. Now I will be okay giving this one. Okay. Okay. What else I want to show you? Okay. This is the case. So I have just given this aptamer situation. Aptamer is very, very important. And then since you are making master degree, you know, that you have to know about this master, aptamers. Aptamers is DNA structure, can bind the protein. So it means that you can detect the protein. It is something artificial antigen antibody interaction, you know. Okay, it means that the aptamer can recognize the protein, you know. By this way we can apply this one. Well, if you want to continue on this this technology CRISPR, you can write me and then I can send you some documents if you want on this area. But if you wish it, you know. Okay, if you wish, I can send and you can ask me any question that you can ask me. So, okay, now the time to sharing you with the questions. Let me go to, okay. So where is okay now? Okay. Expand. Okay. Okay. Yes. Hello. Okay. Okay. So, well, is there any questions that you can ask me? Okay, so I will make the outline what we have discussed today. Okay, so let me open it finally. All your stuff. Option. Okay. 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 Sharing makers. Okay. Okay now. So now I will be explaining, what I discussed today. Okay, what today I just explained to you. Let me go from the beginning, from the beginning. Yes. Let me go this one. Okay. We go. Okay. We talk about the Cas enzymes, Cas enzymes, and there were three enzymes that you discussed about it: Cas9, Cas12, and Cas13, Okay, what is the Cas9, Deactivated Cas, If you, as I said, the Cas enzymes, Cas9 enzymes, two domains, two parts. If you mutate one part of it, and then you can get, mutated Cas9. So it means that it cannot cut it, but what does it do, It can combine, it can combine the target DNA. Okay, so we discussed about guide RNA, single guide RNA. What is the difference between this guide RNA and single guide RNA? If there is in Cas9, there was two RNA, and when they join together, by chemically, then is single guide RNA. And double strand breaks. And what is the big difference between the defense mechanism and gene editing? In defense mechanism, you can kill down the virus, but in the gene editing, when you make double strand breaks, they want to join together. When they want to join together, they will not join the same place. And then what will happen that in this case, the gene will be a different gene, and then the protein will not be produced. In this case, what we call it knockout. I think this last sentence is very important. Now, please remember that. What is the knockout or gene, Okay, when you make a double strand breaks, when double strand breaks by guide RNA, when they want to join the DNA again, but they will not join from the same place. So it means that the gene that you cut it down has been killed down. And then what's we call it knockout. If this gene is mutated, that you eliminated this gene, okay? You eliminated this gene. This is very, very important, okay? So one of the main items that I explained it, knockout a gene, it means the treatment of the cancer, treatment of a mutation, this is one way this is possible. So what about application of CRISPR to plants, For instance, let me give an example, tomato, What can you do with the tomato, Normally you buy tomato, it's ripening in 1 or 2 days, okay, depending on the temperature. But if you block a gene that makes the ripening, there is a gene in tomato ripening gene. If you kill down this gene, then it means that by the CRISPR technology, you will get what you get, tomato that will never ripen, okay? It will stay as it is the original, This is how can you apply this technology in food science, okay? This is also very, very important, in this technology, not only using the for the health science, but it can be used in the food science. But one thing that I would like to give you one example, for instance, the genetically modified organisms. You know that genetically modified organism that is food, for instance, that you add different genes to the food, but in the case of CRISPR, you are not adding any gene to the system. Okay, what you do, you just cut it down the gene. Okay, the difference between the CRISPR technology and the genetically modified organism is this one. That's also possible that you can use this one, this technology. Okay, now you can use treatment of the diseases, this is okay. At the moment, there are two application that is approved by the FDA. FDA is the United States, the institute that approve or not approve the method. Okay, it's already approved that the sickle cell anemia. This is the genetic disease that is obtained, and the other one is the sight loss. Sight loss. Sight loss means that the patient cannot see because of this disease, So what you do in this case, you you make this gene, mutated. So when you make this one mutated, it will not you produce any protein like this way. So it means that application of the CRISPR in disease, and then also for the treatment of the cancer, that's also possible, and also possible for the agriculture. Agriculture is also a good way to eat it. And what else we have discussed then? Okay, and then this is the first paper came out by the Ishinowa, and please remember that this is very, very important, Ishinowa. And this is palindromic repeats. Let me show this. Okay. Okay, so this is the palindromic repeats, okay, as you could see that. And then it's always the loop form, loop form. And then what else we can see more? Okay, this is the Cas9 and Cas12 and Cas13, But in the case of Cas13, this is the, enzyme can apply to RNA, Okay, now what you have learned more about in this system, Cas12 works on the DNA, but Cas13 works on the RNA, okay? For instance, coronavirus, this is RNA, can be applied for Cas13A, okay? So, okay, this was the story of the Moika, okay, Moika, and please go on this paper if you want, have time, And what else I want to show you is this case. Okay. And then, as you know, that the central dogma, DNA, RNA, and the most important, the off-target, off-target. And due to this off-target business, treatment for the embryo state is not allowed, okay? It can cut not only one place, but it can cut more than one places. And what else? Okay. So this is what I want to tell you. So aptamer that I have mentioned about it, So I think I want to give you some basic ideas about what I discussed today, the defense mechanism of the bacteria, and then gene editing, okay, gene editing, and the electrochemistry of the electrochemistry, which is important part of the diagnosis, the diagnosis. I don't know whether you have seen, electrochemistry needs some additional courses to learn about it. It's not so simple to learn it at the beginning, but if you want to study, on electrochemistry, you must go to, in this area. And then what else? So, okay, now let me see how many people at the moment. Okay. Hey, Tapa, Sankilia, Sana. Okay, Red. Hello, Red. Okay, Red. Do you hear me, Red? Al Sadi.
Yeah, Doctor.
Okay, okay. Do you have any question, or can I ask any question to you? Yes. What you have got, Okay. So, what do you learn about the CRISPR? Tell me something, a few words. Okay. What is CRISPR? Or okay.
About general CRISPR?
Yeah.
CRISPR, this application used the diagnosis the disease, the diagnosis bacterial, genetic, and uses the development, some research about the culture. About now, some country use the CRISPR diagnosis the virus. For example, the Emirates use the CRISPR diagnosis the the COVID-19.
Yes.
Because this application need now development, more development. Use the speed diagnosis the disease bacterial and virus and genetic. Other use can use the treatment same this disease.
Okay, this is good. Thank you. And then, do you have any idea about aptamers? Aptamer is
Okay, doctor.
Aptamer is a DNA. Is bind
Okay.
to the protein. Aptamer. Okay. Okay. So, what else? Okay. So, if you don't have so you can go to my slides, and then you have already a copy of it. And if you have any question, you can ask me. Okay. Okay. So, what else I want to say more about to you? Okay.
Okay,
Because
doctor.
yes. Okay. Thank you very much. What about another student? Song Li Yali. Song Yali. Hello, Song. How are you?
Hello, Doctor. I'm good.
Do you have any question, or what you have learned from this course,
I have no question.
Okay. Tapa. Hello, Tapa.
Yes, Doctor.
Okay. Do you have a question about this? Okay. The CRISPR.
No,
Okay. Now, this is really a mixture of the all science, maybe electrochemistry, biosensors, and then other topics together, Let me show you if I have some other thing to show you more. Okay. For instance, not this one. Okay. Mhm. So, do you see my presentation now? DNA, RNA? Yes? At the moment?
Yes, Dr. Yes.
Yeah,
Yes,
this is
Dr.
very, this is the, for instance, the nucleotide, the other nucleotide, they join together, by the phosphatase star bond, okay? And then the enzymes cut down here, okay? This is enzymes cut here, okay? This is very important. That's why I put this one here, very, very important, very important. This is the 1', 2', 3', 4', 5', and this is the phosphate group. That's all, And then if there is no oxygen in two positions, it's called it, DNA. And if there is oxygen, it's called RNA, Okay. And then what else? Okay. This is what I did. Okay. Shinoa. Okay, this is the scientist that, the Francis Moika, okay. What he did, discovered repeats in archaea, okay. And okay, this is the Spanish scientist. And what else? Okay. Okay, now this is the Barango. Barango is okay. Barango is the first paper, come out. And then if you want to go more about this one, he is the author of the Journal of CRISPR Journal. Okay. What else I will show you? Okay. Okay. Okay, this is all about this one. Now let me show you another slides. Okay. Well, one thing that I really want to show you that really is very interesting in CRISPR technology. I will be giving you, let me show you, is this one. Okay. Yeah. Yes. Again, CRISPR business, when this, this is the virus, red virus, it is the part of the virus has been cut it down and put it here, Okay, one question is this one. Is this cutting business randomly or definitely? Okay, any question? This is a big question, Well, this is the DNA of the virus, When the first time came to the bacteria, bacteria cuts part of this DNA and put it here, Okay, question. Is this randomly cut it down or at a fixed point? What are you talking about? What is your idea? When the first time the virus came to the bacteria cuts part of the DNA and put it in the CRISPR locus. I'm asking the question.
It is. It's not random.
Random or not random?
Not random.
Thank you very much. Very good. Very good. Thank you. It's very good because it is cut it down next to PAM sequence. This is very important. That's very nice. Thank you very much. Very good. It's one of the important questions, this one. And this is the bacteria genome, in bacteria genome. What is in bacteria genome, There are repeats, black repeats, and the colored is the fragment of the virus, different virus came to it, and these are the questions, later, these are going to pre-CRISPR RNA and then finally getting the CRISPR RNA, and then the Cas genes, is becoming the protein, okay? As you remember that DNA goes to RNA and then the protein. So these are going to protein, okay. So this is this is another way of showing the system. Okay, this is the CRISPR array, repeats spacer. Okay, repeats are the same, but spaces are not the same. Okay, as you could see that the Cas1 and Cas2 enzymes, cut the DNA of the virus and put it here, okay. This is there are many ways to show it, okay. I will be showing that if possible, okay. And I think this is the Cas9 and the guide RNA and the matching sequence. Okay, so it means that, for instance, you are a scientist, okay. You want have a knockout, okay, in your system, knockout. So what you should have done first of all, okay? This is a good question, and please try to understand, and I will help you if you do not answer the question. And you have a gene, okay? Say X gene. Do you know the sequence of it? Yes. Okay. And this is a problem, it's producing false protein, So how can you make this gene knockout? What you need to, first of all, you need to design guide RNA. You agree? This is guide RNA. This is guide RNA. You have to design the guide RNA. It means that you will be searching a part of the gene that, DNA, and this DNA complementary to the DNA RNA, First of all, you will select the DNA part of it, a part, when you select the DNA part of target, then you will convert this one to RNA, It means that this is your guide RNA. But nowadays, if you want to work on this CRISPR technology, you have a X gene, and you go to company. You are asking the company that you want to make knockout X gene, and give the name of the gene, and then the company will give you the right guide RNA to you. Okay, but if you do not have this facility, what you can do, there are also software which is free, it's one of them is Chop, for instance. You can go to that website and you just write the name of your gene, and then the software will give you the right guide RNA to you. Okay, it means this is also easy, Okay, so in order to make this gene therapy, gene technology, and CRISPR technology, it will be better to discuss between yourself, because alone this is not possible. You just discuss together, This will help you more important things. Okay, now, okay. Well, for instance, this is application of the SARS-CoV detection, This is also possible by this technology. Okay. So now I talk about the CRISPR, but nowadays is exceptionally important thing that I will give you, anti-CRISPR. Okay, So now, defense mechanism of bacteria, okay, we do understand it. What about the defense mechanism of virus, Then in this case, anti-CRISPR. Anti-CRISPR, a defense mechanism of viruses, Please remember, this is a new technology. Put in your mind, but do not mix it, This is okay, this is okay. These new things, okay? That, in order to, for instance, what is the use of anti-CRISPR, So in this case, the attacking is the bacteria, So how can you eliminate the bacteria in food science? This is what we call it anti-CRISPR technology. But this is only just remember these one or two words. And then what else I can show you more in my presentation. That's more about electrochemistry. This is the university that I collaborate, This is university in Cyprus, Lancaster University, I've been, and then the universities in Malaysia, the Ege University, and this is the other university, and these are my collaborators, collaborators. I have a student from maybe Nigeria, Nigeria, and there is Fadi Al-Turjman. This is also a professor in our university who is working on the artificial intelligence. Okay. What else? Okay, there are mostly the different countries. Well, this is another application of the CRISPR technology. Artificial intelligence, artificial intelligence. This is the application. And that's all what I want to give you more because in this case, so lastly, what I can say more that since we have a lot of things covered, that spacers and what we call protospacer, that is we discussed and that. And then what else I can give you? Okay. Now it's time to leave. Maybe if you don't have any questions, okay, we can stop here. And then I'm open to any kind of question that you may ask, okay, for future benefits, electrochemistry and other things. Okay. 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 Read Zadi Sani Songlai Tapa. Okay. Okay. Well, if you want, you can add by yourself any other things that you know about the CRISPR. I will be happy also. If you want to add more things. Yes. I hope that.
Thank you. Yeah, Prof.
Yeah.
Thank you very much. I think students, do you have any question?
Any comments?
Any
No.
comments?
No.
Okay.
No comments. Okay. But anyway, the class by Prof is already recorded here. So I will pass the recordings to everybody in case you have any question or comment to Prof. You can directly send it to Prof Mehmet, or if you have any question regarding the topic, is it going to be assessed under certain assessment, you can ask me lah. Okay. With that, I think, Prof, thank you very much for today. 2 hours of session. I went out because I have a discussion with Azia just now. And then I told her, Azia, I need to go back to the class because Prof is teaching.
Okay, give my best regards to Asia.
Yes,
Okay.
yes.
Okay.
Yes,
Goodbye.
definitely, inshallah. Okay, Prof. Take care.
Take care. Take care.
Okay, bye. Thank you.
Thank you.
Bye.
Thank you, bro. Thank you.
Welcome. Welcome.
Bye.